Hi everyone, my name is Fleur Ponton. I'm from the School of Natural Sciences, Macquarie University in Australia. And today I'm going to talk about my work on nutrition, infection and symbionts. First of all, I would like to acknowledge the traditional custodians of the Macquarie University land where this presentation was made, the Watamatagal clan of the Darul Nation, whose cultures and customs have nurtured and continue to nurture this land since the dream time. We pay our respects to elders past, present, and future. Host-symbiont interactions are, I think, one of the most fascinating phenomena in biology. Nearly all organisms on this planet have their body inhabited by other organisms. It could be parasites, mutualists, or commensals. I'm also fascinated by nutrition and food. So I don't know if it is because I'm French or because I grew up in the restaurant of my parents' restaurant, or if it is because nutrition is essential to so many physiological functions. So we all know that nutrition interacts with longevity, aging, development, reproduction, but also immune responses, and foods are sources of symbionts. So how to study host nutrition is actually not an easy question because foods come packaged together as mixtures. So if you take those snow beans, for instance, they are composed of 83% of carbohydrates, 14% protein, some fat, some minerals. And so there is a balance of nutrients that is already packaged in this food. So this is the approach I will have in all the studies I'm going to show later. I won't study nutrients separately, but food as a mixture of nutrients. And I mainly work on insects. So what are the main nutrients for insects? Protein and carbohydrates are actually the major yielding energy nutrients. So all my studies, the one I'm going to be showing, are about the balance of protein and carbohydrates in the diet, and also called the PC ratio. So the first question I was interested in was to understand if infected hosts show a modified feeding behavior and why they're doing so. The first model I used to approach this question was the association between Tenebrio molitor, this little cute black beetle, and the tapeworm Hymeneleptis diminuta. So Hymeneleptis diminuta has a complex life cycle that starts with the insect, but I won't go into details for this life cycle. So we had infected and non-infected beetles, and we gave them food choices, so different type of food, and they had a choice between two of these food. All this food varied in the protein to carbohydrate ratio. Some were biased in carbs and some were biased in protein. And we measured their food intake, fecundity, and lipid reserves. So it was quite interesting to see that the infected beetles in black on this graph injected different amount of carbohydrates when they could do so. So here you can see choice 1 and 2. They increase their carbohydrate intake. Choice 3, they don't, and certainly because they couldn't. It means that they were reaching the maximum amount of food they could eat, and this wasn't translated in more carbohydrate. So when they were able actually to increase their carbohydrate intake, they do not suffer any negative effect on their fecundity anymore. What was observed when they couldn't actually increase their carbohydrate intake. So it was quite interesting to see that beetles infected with the tapeworm could actually shift their diet choice when they had a choice, and this helped them to decrease the negative effect of the infection on their reproduction. It was a bit tough to work with this tapeworm because we needed to have the full cycle in the lab, and was a bit of a nightmare. So I shifted my work on Drosophila melanogaster, and I first used the bacterium Micrococcus luteus. Flies were infected, so we had naive flies, we had flies infected by Micrococcus luteus, and we also had flies that were sham infected, so we just injected them with PBS. And we did similar work that with the beetles before. we gave the flies, in this case, a choice between a food that was pure sugar and one that was pure yeast. And we measured their food intake every 2 days for 6 days. So on this graph, you can see in blue the sham-infected flies, so the nutritional feeding behavior of nutritional choice, let's say, of the sham-infected flies, the naive flies, and in orange you have the infected flies. So the infected flies decrease their intake of protein, which is quite clear here, but they maintain their intake of carbohydrate, which means that they were ingested food that was carbohydrate biased. When we fed the flies with a single diet and we look at their survival after infection, it was quite fun to see that the flies, when they were infected and fed the high-carb diet deselected in the experiment that was done before, they actually survived better the infection than when they were fed the balanced diet that was chosen by the naive and sham-infected flies, or a low-carb diet that for all treatments we saw a very bad survival. When we looked at the naive flies, and it was similar for the sham-infected flies, we're finding that either the high-carb or balanced diet, they survive quite well. So it really seems that by composing high-carb diet, infected flies increase their chances of surviving the infection. We also followed the expression of antimicrobial peptides for these different diets. We had actually 8 diets in this case that vary in the percentage of protein, dietary protein, and the percentage of carbohydrate as well. So when the percentage of protein was high, the percentage of carbohydrate was low. And we found that there was a negative effect of protein on the expression level of antimicrobials. So it really seems that by selecting a high-carb diet, the flies might be stimulating the expression of their antimicrobials, and this might help them to better resist infections. We did similar studies using another fly, which is Bactrocera trioni, or the Queensland fruit fly. It's a major pest in Australia, infecting lots of fruits, so it's quite a good model to work on. And we use, in this case, the bacterium Serratia marcescens. We had flies infected by the live bacteria with heat-kill bacteria, also injected with PBS and naive flies. And we had a very, very similar protocol than the one I just showed you. We also found, in this case, that flies, when they were infected with the live bacterium, they shifted their diet, they decreased their intake of macronutrient, and ingested a diet that was carbohydrate biased. When we looked at the CFU, the number of bacteria in the body of flies that were maintained on the single diet that they selected, we find that the infected flies maintained on the high-carb diet seems to have less bacteria in their body than the one that were maintained on the diet, selected by the naive, heat-killed, infected, and sham-infected. So again, we see that this high-carb diet might actually help the host to fight the infection or to maintain the growth of pathogen lower. So where we did this work during her PhD, she was also very interested in transgenerational effects. So she manipulated the diet of the moms, the dads, and looked at if she could find a similar effect in the offspring. So she looked at a lot of developmental traits, but also the adult offspring survival after infection. She did not find much, actually, which was a little bit disappointing, considering that it was a very large amount of work. But she found that there is an effect of paternal diet on the son's resistance to infection. But in this case, she did not find the effect of a high-carb diet, but more a balanced diet. So this is something that should be continued and explored again. Okay, so this was about infections, but I'm quite interested in my lab about symbionts as well. And we had a look at how symbionts can influence the host foraging for bacteria. So it was a study that was done a few years ago by Adam Wang while he was a postdoc at Sydney Uni. And we looked if manipulation of the gut microbiota was influencing the foraging of flies when they were offered diet with different types of bacteria. All of them were found in their digestive tract. And we indeed found that actually when the flies were conventional, so naive, not touched, they choose diets that contain core gut microbiota, Lactobacillus plantarum and Ascicter pomorum. When they were axenic, they were only going for the food containing Lactobacillus plantarum, and they didn't want the food with Ascicter pomorum. So it seems that there is an innate attraction for Lactobacillus plantarum. And then we explore the foraging of flies that were mono-infected by these two core bacteria. And it seems that Acetobacter pomorum, when it's present in the gut, overrides the preference for Lactobacillus plantarum, which is quite interesting actually, because the flies with Lactobacillus plantarum behave exactly like the conventional one, was the one with Acetobacter, when there is an imbalance in the gut, they actually go on food containing Acetobacter pomorum. And we know that this imbalance in the gut, it was another study that we did completely independently, does influence a lot fecundity in a negative way. So it's quite interesting that if you have an imbalance of bacteria in the gut, or flies have an imbalance of bacteria in their gut, they have different foraging, and this foraging might maintain this imbalance, and in this case, a low fecundity. We also look at the effect of symbionts on host foraging for nutrients. This was a work done by Bean during her PhD as well. She also treated at axenic flies that were controlled or re-inoculated by bacteria, and looked if they were ingested different amount of carbohydrates or proteins. She found not so much effect actually, except there was a trend, but it's a trend of females, axenic females, increasing their intake of carbohydrate, while the male, it wasn't that obvious. Despite having this different, this trend, the axenic flies still suffer quite a lot negative effect of the manipulation of their gut microbiota. Okay, so it was a bit fast, but this is what we're trying to explore in the lab, all this interaction between host diet, microbial community in the gut, infection. We still have a lot of work to do, but this is a fascinating field of research. All this research was supported by the Australian government, Macquarie University. We're working quite a lot on bees at the moment, and there's a lot of work that's going to come out for honeybees and stingless bees. I would like to thank all my collaborators and colleagues, and here you can see some of the people in my lab. Thank you very much for your attention.